Hello, welcome to the first tutorial on using bioinformatics tools in further education genetics teaching. What we're going to look at in this first video is how to use databases that contain genetic and protein information, how to find your way around them. We're going to search for two very specific genes and we're going to find their sequences before we manipulate them in the second tutorial. We're also going to have a look to how to use different shortcuts on keyboards, which will be helpful for you in the future. It's useful when you are doing bioinformatics, if you have a blank text edit document open, it's better to use text edit, not to use Microsoft Word, because Microsoft Word likes to try and correct little things that you have in your sequences, and we don't want that to happen. We just want standard text format, so text edit is good. So what we're going to look at today is what we call Hox gene sequences. Hox genes are really useful for us to study in bioinformatics because we have many genes and their sequences are slightly similar, which means that we can compare them. They're slightly similar because they have evolved from one another. Before we go any further, we're just going to go and have a look to see how we can use our different shortcuts. So first off, you are going to be highlighting quite a lot of things. So the best way to highlight is to do Control A. So you can see that on my little keyboard at the bottom right there. If you want to copy something, then you want to go Control C. If you then want to paste something again, Control V. Nice and simple. That saves you from just having to use your mouse and to highlight sometimes thousands and thousands of sequences that you're looking at, so thousands of letters at a time. So the first database that I'm going to show you is one called NCBI. It's the first one that comes up. Now, NCBI is a really, really great site that has a lot of information, way more information than you will possibly need. It works exactly like Google. It has lots of different databases. We are looking at specifically genes, so we are going to go to the gene database. If you just wanted to look for generally what was happening, then you could just click all databases and it would show you everything. So I'm going to search for a gene called HOXA1. HOXA1 I'm going to compare later to HOXB1 because it's the one that's really, really similar to it. We have lots of options, which we are not going to look at today for reasons of just keeping what we're doing at the moment as simple as possible. You'll note on the right-hand side here, you have different organisms. Now for us, we're going to look at Homo sapiens, which is the first one that actually shows here. However, if you were doing other types of research, then you might want to specify that you are looking at Xenopus laevis, which is a toad, or Mus musculus, which is a mouse. So I'm going to click on HOXA1, and it's going to bring me to this really massive page that has a lot of information. I'm only going to show you a few bits of the information here, just because for time's sakes. It shows you the chromosomal location, so it tells you it's on chromosome 7. It has lots of bits that we're not too worried about at the moment, but these are transcript variants. You will look at them more when you go to your degrees. It tells you where you find the specific protein for HOXA1, what it becomes. There's lots of references that you could look at and so on and so forth. That's lovely, but what we're interested in today is specifically the sequence. So what I'm going to do is I'm going to come to the right-hand side here where it says related information. I'm going to scroll down until I find nucleotide. I'm going to click on nucleotide. And what that's going to show me is a list of all the different nucleotide sequences that are associated with HOXA1. It's actually a really long list. It's way more than I actually want to look at. Sometimes it's just something that contains HOXA1. So for example, on query number 4 here, it's actually the entirety of chromosome 7. That shows me that result just because HOXA1 is on chromosome 7. The best way to have a look at this is just experience, there really is no better way to do that. I'm specifically going to go to transcript variant 1 right now, just to show you guys what we're looking at. So if I click on HOXA1, it gives me some parameters about, the nucleotide, how it was made. I'm going to ignore all of that, and I'm going to come down to the bottom, and it's going to give me my sequence. Now that's really nice, but if I just copy and paste that for a second, I'm going to do it the old-fashioned way of highlighting. So I'm going to copy, I'm going to paste it over here. I now have all the numbers here, which is fine, but it's not what I want. I've also got gaps everywhere. And for when I want to actually manipulate that later, that's not going to be useful for me. So I'm actually going to get rid of that. I'm not going to use my sequence like that. What I'm going to do is I'm going to come back over to NCBI, and I'm going to click on this, FASTA. FASTA is a format for how we look at genetic and protein information. Now this is much nicer. All of my nucleotides are together. I have a little title. It's going to tell me what that is. And I've got no gaps. So I'm going to highlight that, and I'm going to paste that into my text edit for later. Now I'm not really happy with what this is being called right now. It's a bit long, so I'm going to keep the chevron, and I'm going to call that HOX A1, and I'm going to put full. This is going to help me later, because hopefully by this point, you know that 3 individual nucleotides together make a codon, and the first codon of any gene is always methionine, ATG. My first codon here is not ATG, it's ATC. That is telling me that it's not the start of my gene. My start of my gene is actually after this point. Now, if I go back to my search, you note that my gene is actually quite long here, so 2,500 base pairs. So I'm going to go through the list, and I'm going to see if I can find the better version of my gene. And if I scroll down, you have quite a lot here, if you notice. I'm going to come to ones like this. See where it says cDNA? See if there's a better one. There we go. So at the bottom here, I've got HOX A1 protein, mRNA complete CDS. Now that's telling me that is my complete sequence without introns. So it's only exons. So I'm going to do the shortcut this time. I'm going to go to FASTA. And you notice now my sequence is much shorter. You'll also notice that it starts with ATG. So I want that. I want that sequence. So I'm going to copy that. I'm going to come over here. I'm going to paste it in. And this time I'm going to call it HOX A1 exons. So it only contains my exons. We're actually going to get rid of the space there, because that's going to help us later. And that is it. What I'd like you to do after this tutorial is see if you can play the video back, find your own HOX A1 gene, your own HOX A1 nucleotide, copy and paste it into your own text edit. What I would then like you to do is to find HOX B1. We are going to need HOX B1 for our next video. I hope that you enjoyed this video. I hope that it was informative for you. And I will see you in the next tutorial.